There should be a countdown. We're live. Hey, okay. Well, welcome to our live stream. Wow, we already have 10 people here. So, greetings. Thank you for coming. This is the Cohen Center for Recovery from Complex Chronic Illness at the Icahn School of Medicine at Mount Sinai. And we are leading a conversation about roots and drivers of complex chronic illness. And today's topic is autoimmunity. And so, if you saw the illustration, the cartoon illustration that we put up, and we were asking if you could guess what the illustration was demonstrating, the correct answer was autoimmunity. And thank you to Matteo Farinella, who made that beautiful illustration. And so, yes, we are I'm excited to have you today. If you're watching, let us know where you're watching from. If you have any questions, let us know in the comments. You should be able to post a question. We are accepting questions to answer about autoimmunity. Okay, so to introduce our co-hosts, I have Amy, Dr. Amy Proel, who is the scientific director at CORE, and Dr. David Petrino, who is the Nash family director of CORE. I am Dr. Raven Baxter, and I am the director of science communication at CORE. Great. So without further ado, let's dive right into the topic. Autoimmunity. Amy, David. Autoimmunity, in simple terms, is when we have the body cells attacking itself. So that is generated by different instances. Autoimmune diseases can be acquired in various ways, but in this conversation, we're focusing specifically on autoimmunity as it occurs in infection-associated chronic illnesses. So, Amy, let's further define autoimmunity and IACCs. And moving forward in this conversation, we will be using the letters IACC to talk about infection-associated chronic conditions. So, Amy, can we talk more about the relationship between autoimmunity and IACCs?
Sure. So IACCs, it's just a broad name that we're giving to a phenomenon in which a person gets an infection. So let's say SARS-CoV-2, the driver of COVID, which we're all very familiar with, and then goes on to develop persistent symptoms that sometimes can last a lifetime. This happens with many pathogens, and there's in ME/CFS, it's another chronic condition that we study and treat. There's a history of enteroviruses, viruses that are other respiratory viruses that are associated with many historical cases of ME/CFS. We also study chronic or tick-borne vector-borne illness, which means the chronic consequences of getting infected with Lyme disease or other co-infectious pathogens. And we want to understand in these patients why, after getting an infection, or in some cases multiple infections, they get these chronic symptoms that are often very debilitating. And one reason, obviously, that has been put out there in medicine is that autoimmunity could be part of the reason that chronic symptoms occur after an infection, right? And I think that's autoimmunity typically, so that the historical and sort of most basic definition of autoimmunity is that it's the immune system attacking itself, right? So our body is attacking itself. So in other words, for some reason, and again, that's part of it, is like, why? The biggest question that I've always sought to ask when thinking of studying autoimmunity is the why, but the idea that immune cells, especially potentially our T cells, are attacking our own tissues or our own human receptors, and that, of course, could lead to symptoms. One of the interesting things, though, that I think is important to consider is that the theory of autoimmunity, which is this idea that the body and the immune system can attack itself, was created at a time when the human body was thought to be almost largely sterile. So in other words, it was created before the human microbiome, which is all these, and we've discussed this in previous episodes, we are all filled with ecosystems of interacting organisms, bacterial, fungal, even phage organisms, these huge interacting communities of complex organisms that are in many body sites, the gut, the mouth, but also in other sites. And we're increasingly understood as humans to also harbor what's often referred to as the human virome, which are viruses, and sometimes actually bacterial pathogens are part of this, or fungal pathogens as well, actually, or parasites. So really, it's basically like a pathobiome type of different pathogens. But even in the womb and across the course of our lifetimes, we acquire, for example, the herpes viruses, like Epstein-Barr virus, herpes simplex virus 1 or 2, cytomegalovirus. Also, some people acquire parasites like toxoplasma. So all as we're going through the course of our lifetime, we also become host to these different pathogens, which we've discussed in previous episodes, can often be in a dormant form where they don't seem to drive disease, but they can also activate. And when they do, they create proteins that can be part of disease processes. And so the first thing to think about is that the theory of autoimmunity was developed when we didn't know any of that. And I think that's important because the theory of autoimmunity gained a lot of hold with this idea that the immune system just goes a little crazy and attacks itself, because there wasn't really any other explanation at the time. This was like maybe the 1960s, 70s, where the theory of autoimmunity was gaining hold. If someone got chronic symptoms and we could recognize that the immune system was targeting something human, a part of self or a human receptor, there was no other explanation than it's got to be something wrong with the immune system. The immune system has to have flipped out or become dysregulated. There wasn't the possibility, which is what my favorite thing to study in autoimmunity is, which is to consider, could some of these infections, whether being the single infections like the herpes virus infections or the collective activity of the organisms in the human microbiome, could they be at the root of sometimes why the immune system becomes dysregulated and attacks self? And I can get into that later, but the answer is yes. In some instances, they absolutely are. For example, via a phenomenon called molecular mimicry that I can explain in a little bit, where a pathogen, a protein created by a virus, for example, is really similar in size and shape to a protein that's part of our human pathways. And when the immune system targets the viral protein, it cross-reacts and hits our human protein as collateral damage. And that means, in that case, that the pathogen was actually at the root of what was called, but previously would have been deemed sterile autoimmunity. So that's a little bit of where I think we're at with this, is that autoimmunity is very likely happening in some patients with long COVID or ME/CFS or complications, symptoms like after chronic Lyme disease. But again, the question is why? What are the mechanisms underlying autoimmunity? That's what I think is interesting to most explore.
Yeah. I think that was awesome. I would just add to that, just the fact that there is this sort of concept of the mosaic of autoimmunity. You will probably read about this if you dig into autoimmunity and its role, and that is a theory that is constantly being updated, which tells us that when autoimmunity is present, so when the body is seeming to attack its own tissues, I think a lot of folks who are studying autoimmunity will acknowledge that the reason for that is a combination of persistent pathogens, genetic variables that may make somebody's body more susceptible to an overactive immune system or a dysregulated immune system that is engaging in activities of this nature. And then also environmental factors that, can really affect a human's immune system and cause them to, cause their body to start becoming highly reactive to certain things. So exposure to chemicals, exposure to stresses, exposure to all sorts of different things in the day-to-day environment. And so I think that as we delve into autoimmunity, that keeping in mind that model of the mosaic of it is undeniable that your body is currently attacking itself. Now let's figure out what is contributing to all of these things that is leading to the body to attack itself becomes the next important step, as opposed to, I'm just going to slap this diagnosis on you and suppress your immune system because your immune system's too active. That is the lens that we need to get out of, because it's potentially catastrophic for people who have one part of the mosaic overrepresenting versus other parts.
Thank you, Amy and David. And so autoimmunity is not anything new to the medical community, and so with that being said, we do have treatments that exist for autoimmune diseases and conditions. What current research, and I know the answer to this, but can you shine light on the current research that draws from current research in advancing our treatments of autoimmune conditions as it relates to IACCs that is being pulled from our already existing fund of knowledge about how to address autoimmune diseases? Amy.
Okay, sure. I mean, look, I think that we actually need to be careful with the kinds of therapeutics that we bring into this space. Again, when we are studying autoimmunity in patients with long COVID or ME/CFS or other conditions, because it's really important to understand that we're at a time in history where conditions that have been deemed autoimmune, sort of in the most generic sense, let's say like lupus or multiple sclerosis, that are normally the standard of care for them now is immunosuppressive drugs that you have to take for your whole life. You can't get off them because in a sense, like they're more palliating the issue than they're not curing you. They're just knocking down the immune system, right? These classic, if you will, autoimmune conditions, infection is actually being placed at the actual root of their pathogenesis. And so it's important as we move forward to understand that, for example, in multiple sclerosis, there was a really incredible study by a team at Stanford where they were able to take cerebral spinal fluid from patients with multiple sclerosis, isolate antibodies, and show that there's this basically neuron protein in the brain that's part of our human self that's targeted, that becomes part of MS symptoms by the immune system in the brain. But they showed for the first time ever that there's an Epstein-Barr virus protein that has a similar size and shape to that human neuron receptor that gets targeted in the MS brain. And again, that it was the immune system firing on that Epstein-Barr virus protein, and then this crossfire onto the neuron protein that really might be at the heart of the autoimmunity in MS. And so that's leading in MS to a revolution in the way the MS community is starting to approach treatment. Because for now, yes, patients are put on immunosuppressive drugs that just kind of knock down their immune system with the hope that if you knock down the immune system, then the immune, there'll be less inflammation as part of this and the symptoms will improve. But there's also groups that are actually bringing in treatments for Epstein-Barr virus into the space, right? Because maybe, and of course, if you could just get at the virus that the immune system is going after as part of that leads to that cross-reactive reaction, that could potentially sort of get to the root of autoimmunity. And that's even where we got part of the collaboration where we're running at CORE now. We're getting a trial of the antiviral Truvada started in patients with long COVID. And we're trialing it for multiple reasons. We think there's maybe a chance that Truvada could affect SARS-CoV-2. SARS-CoV-2 persistence is definitely an issue in long COVID patients, but also in MS, there's a team, at least one team, trialing Truvada as a possible way to mitigate Epstein-Barr virus activity in multiple sclerosis, because some studies have shown that it could have activity. So you have this world where, yes, if you get this autoimmune condition, multiple sclerosis, and you go to the average doctor, you're going to be put on immunosuppressants. But if you look at the most cutting edge research trials, they're actually going after the viruses and that are starting to be put at the root of this cross-reactive autoimmunity. So I think that in our sphere of infection-associated chronic disease, we definitely need to think that through. Well, we could potentially put patients on immunosuppressive drugs for, but I think that what we really need a pretty high burden of evidence. Number one, like, we really have to know that the patient has an autoimmune issue or a clear autoantibody and a functional autoantibody. Maybe David can describe that a little bit in a bit, because autoantibodies are often generated really transiently, often in just human disease. For example, when someone gets a new infection, the act of the immune system going after the new virus or the new bacteria leads to a lot of immune cell activation and all kinds of cells getting involved. And that's a great time where there's a lot of opportunity for quick cross-reactivity between different organisms and different immune cells and autoantibody generation, right? But a lot of those times, those autoantibodies generated there live for a bit and then go away, right? So you have to do, and David, you could describe the work you've done with the Kiko in mice, where you did extra work to take some, in this case of one long COVID study, to actually make sure that autoantibodies that were isolated from long COVID blood and then injected into mice, David could talk about this study, actually really were able to drive disease or symptoms in a mouse, and giving them more sort of clout as actual agents of problems than some of the transient type autoantibodies that can be created. You have to be careful with that, because you don't want to overtreat based on that. So I definitely think we need to have really clear evidence that someone has an autoantibody issue happening. We don't just want to say, because this has happened, long COVID might be autoimmune. And you know what? Then we should just maybe give someone an immunosuppressive drug because, hey, we're not sure what else to do. Probably that's not thinking I would support, because the truth is that putting someone on an immunosuppressive drug might make them feel better for a while, because of course it could knock down inflammation. But the issue is, again, if infection or even microbiome issues could be at the root of some of the person's problems, knocking down the immune system gives those pathogens or the microbiome itself a really good opportunity to go out of balance. And what kind of happens in other diseases is that patients then often require higher doses of immunosuppressive drugs over time. It's like they go back to the doctor, and a year later, they don't just need the immunosuppressive drug they were put on the first time. Now they need a higher dose, and they need a second immunosuppressive. So people end up getting on to higher and higher doses of the drugs, potentially because these root cause problems tied to infection or microbiome aren't being addressed. And I think we really don't want to do that in patients with long COVID or related conditions without really knowing that the immunosuppression would be beneficial in some other very clear way, especially last, because if you are knocking down the immune system with immunosuppressive drugs, you're making the person more susceptible to other infections too. And of course, if you're a long COVID patient or anyone, you're already paranoid about getting COVID, well, justifiably paranoid or worried about getting reinfected with COVID or any other infection. Everyone gets worse often when they get other infections. So we don't want to knock the immune system down again and make people more susceptible to infection, which is usually, if you look at most immunosuppressive drugs, the first side effect that you'll see is increased risk of infection, right? So that's got to be kept in mind if we're going to prescribe that kind of therapeutic. But David, there are instances where there might be a more specific, really clear example of autoimmunity happening in a patient, let's say with long COVID. And I think under those conditions, there are more targeted therapies. You could potentially, David, discuss the study with the Kiko that I mentioned before. And also, I think some of the therapeutics you guys are considering, or we are even, like the FcRn type inhibitors.
Yeah, I mean, to sort of, again, piggyback on some of the stuff that Amy's saying, I think, it's really important. And I think that this is something that is a common theme in these meetings that we have and these conversations that we have, is that, everyone's approach to treating illness, it tends to be make the diagnosis, and then for this diagnosis, there's this one treatment and away you go. And I think with infection-associated chronic conditions, the biggest issue that we face is this very simplistic method of addressing, illness. is not going to play well because, well, like we always call these things complex chronic illnesses, and the clue is in the title. These are complex illnesses. So, to Amy's point, it is very true that in a percentage of people with long COVID, for instance, let's talk about long COVID for just a minute, we see evidence of autoimmunity. And now, when we talk about autoimmunity, what we're really talking about is, again, to redefine, it's, this parts of the immune system attacking the body's tissues. So that is happening. We're seeing evidence of that. We can confirm that that is happening. However, it could be happening for a number of different reasons. So, like two main, mechanisms of autoimmunity, for instance, are one, which is, one that Amy's already mentioned, molecular mimicry, meaning, and, in a very brief definition of that, we'll just say molecular mimicry is kind of what happens when a pathogen that is present in the body is able to trick the body into activating T and B cells that are designed to attack the body's tissues. Because sometimes we want our immune system to break down our own tissues. And so the pathogen resembles closely enough to, body's tissues that the body says, oh, I think what I need to do is attack the tissue right now. And these autoreactive, that's what they're called, autoreactive T and B cells, go and attack the tissue, even though it's actually a pathogen that is causing that reaction to occur. So we have that. We have an autoimmune, action occurring, but it's because a pathogen is there. You also have things like bystander activation, which is when, bystander activation occurs when you start to basically create an immune response, even though the pathogen isn't there. So, there are ways that an autoimmune response can occur with a pathogen present, and ways that an autoimmune response can happen when the pathogen isn't present. And so, our job at this point is to start to do the research that is necessary to say, okay, given that we're seeing autoimmune activity happening in your body, let's try and understand what the cause is. Let's try and understand why it's happening. And the work that Amy was referencing that we've been doing with in collaboration with Akiko Iwasaki's team at Yale University has been really focused on that, where we took plasma from people with long COVID, we took plasma from people who are healthy controls, and we took plasma from people who we call convalescent controls, meaning they had COVID, they had fully recovered, and and they weren't symptomatic. We took plasma, we isolated IgG, which is an antibody, a class of antibodies that are present in the plasma of these people. We purified that IgG, so we removed any chance that there were pathogens or anything going on in that IgG, and then we did what's called a passive transfer, where we injected that into mice. And in the mice that received the IgG from the purified IgG from people with long COVID, we saw symptoms immediately start to emerge. So we started to see balance issues, we started to see pain issues, we started to see issues with generating muscle strength. All of these issues that indicated that at least a percentage of the IgG that was purified and taken from people with long COVID was capable of then creating these, autoimmune responses and effects in animals that turned out to be damaging. Now, that tells us that there is this presence of functional autoantibodies in the IgG. That tells us that if we were to perhaps in the population of people with long COVID that could cause these symptoms in mice, if we could apply a drug like an FcRn inhibitor, which is a very specific drug that actually, inactivates functional autoantibodies rather than completely suppressing the entire immune system. So it's kind of like a precision strike versus just carpet bombing a whole area.
Mhm.
This could indeed, help folks who are experiencing this form of autoimmunity, which could be causing their symptoms without necessarily compromising their whole immune system. So I've said a lot of words just there, but, to very quickly summarize, we're still not we're not asking, in my opinion, we're not asking detail-oriented enough questions to address the complexity of complex chronic illness. So, what I would urge the people listening to this to beware of is someone saying, "Oh, you have autoimmunity," based on some, very basic blood tests. Let's suppress your immune system and see what happens. That is not a evidence-based approach right now for folks with long COVID or other infection-associated chronic conditions. And we desperately need the research to catch up, because the one thing that we can say for sure is that in a percentage of people with IACCs, autoimmunity is occurring. Now we need to ask the deeper question: why is it occurring? Is it occurring because of molecular mimicry? Is it occurring because of bystander activation? Is it occurring because of another mechanism that we don't quite understand yet? And then let's start to provide very targeted drugs or very targeted therapies to address, the specific reason for the autoimmunity.
Thank you both. And I just want to recap a little bit in layman's terms for some of our viewers. I know we have a wide spectrum of people across many disciplines and levels of familiarity with these scientific terms. So if this is elementary to some and not others, just trying to make sure everybody understands. So your body is like a castle, and inside of us we have an army, which is our immune system, and it protects us from invaders like viruses, germs, including bacteria and other things, right? Things that are not natural. Well, we shouldn't say natural, not self, basically, not a part of like our human cells. And so sometimes these invaders, they can wear disguises that look a lot like the friendly cells in your body. And that is called molecular mimicry. It's kind of like if a bad guy dressed like one of your friends to sneak into your castle to join the party, but it's like, actually, that's a bad guy, and everybody's confused because the bad guy is now attacking your friends, your own cells, thinking that they're the invaders. So this can sometimes lead to illnesses. And so, researchers are trying to figure out basically these mechanisms and how we can correct them or fix them. And then we also talked about, so that was molecular mimicry. And then we talked about the bystander activation. And that's like when the superheroes in our bodies are battling the bad guys. So our immune system is kicking the pathogens, beating them up. Sometimes they might knock over a few buildings while trying to catch the villains. I don't know if you all have watched superhero movies, but they're fighting crime, but like oftentimes they're also wrecking the buildings. And I've actually always thought, what's the benefit to having like Superman beat all these villains up if he causes like $5 billion in infrastructure damage to these cities? Anyways, that can happen in your body too. So that means while your immune system is fighting off germs, the defense system might actually accidentally attack some of your healthy cells that are nearby, and that can be considered autoimmunity because your body is essentially attacking self unintentionally. And then with that, again, scientists, researchers are still trying to figure out how to keep the superheroes focused on only the bad guys. So we're still nailing that down. David also brought up FcRn um, inhibitors. And so FcRn, um, imagine if your body.
I can't wait for this. I can't wait for this analogy. Go ahead.
I'm going to do my best. Okay. Imagine if your body had a special delivery service that helps keep important packages, like the packages that are involved that contain things that help our bodies function, like proteins, safe and sound. One of the delivery helpers is called FcRn, and it makes sure that proteins like antibodies stay in your body longer to help fight off germs. But sometimes we need to slow down the delivery service to help, um, I guess, combat or prevent certain health problems. And that is where FcRn inhibitors come in. And they act like traffic lights. So they can tell that delivery service, hey, take a break, maybe not deliver so many of these packages right now. We don't need that. And in the case of antibodies, that making too many antibodies could cause trouble. That happens in some autoimmune diseases. So we can use potentially FcRn inhibitors to help everything keep everything balanced and running smoothly, and that could be an important advantage here in treating infection-associated chronic conditions. We have some questions here in the chat. Okay, so Amy, Ella asked, is there evidence of chronic Bartonella leading to autoimmunity?
Excellent question. So it just hasn't been studied enough. And I think it's one of the things we most need to study. So again, to clarify, when we're talking about molecular mimicry, the main consideration here is that you have, when you get an infection, a T cell becomes activated, for example. And the thing is that, or an antibody, which is part of the adaptive immune system that's targeted to go after a particular pathogen, let's say Bartonella, for example, becomes activated. But the thing is that antibodies have what's called flexible specificity. So they go seek out the target of what they're trying to find, which is usually a protein made by a pathogen, but they're created to recognize anything that's sort of similar to that. In other words, there's a specificity in what they can identify and then will attack, right? And at the same time, that is good in a way because it might allow them to find, different types of related pathogen proteins, but it's a liability because if there's a human protein or a human receptor structure that's sort of similar to that viral or bacterial protein, it's similar enough that that same antibody attacks that as well, right? And so that's the thing, is this flexible nature of antibodies allows for this cross-reactivity that can happen. In the case of Bartonella, it's just an understudied pathogen in the first place. So I think we're reviving, I think you'll see in the world of tick-borne, vector-borne illness, a lot of, hasn't been enough focus on any of the tick-borne, vector-borne pathogens, but Borrelia, the Lyme pathogen, is, if anything, the most studied. And then Bartonella gets sort of tagged along as like, oh yeah, people get Bartonella too. It's not as studied as it could be in terms of its characteristics. So what we have to do is we have to take Bartonella, and this is one of the top things that I think is happening and needs to happen in research, is that we can create databases, and we can even use computers now, where we take the structure of Bartonella or proteins created by Bartonella. We actually know their sizes and shapes and structures. And then we create a database of human receptors or structures and their sizes and shapes. And the computer just starts to match them up. And we can actually say, oh, for example, this dopamine receptor has a similar structure to this Bartonella protein. That's starting to happen a lot more. For example, there's a team that actually just took some of the more common bacteria in the human body. I love this study. It's like 66 of the bacteria that are common in the human microbiome, some of the more common human, bacterial pathogens like E. coli. And they did exactly that. They took all of the proteins that they could make, and then they took all of these human structures and receptors and just were like, hey, what hits what, right? And the interesting part, one of the things that I find fascinating in that study is they found hundreds of mimics. So in other words, like hundreds of instances in which the bacterial proteins were really similar to the human structures and proteins. And some of the most common targets of the human proteins that were similar to the bacterial proteins were collagen proteins, extracellular matrix proteins, and proteins associated with connective tissue. So there's like this insane propensity for molecular mimicry of organisms to contribute to connective tissue disease, which is something that I think is extremely understudied. Going back to Bartonella, I think that undoubtedly in PANS pandas, for example, PANS pandas is a condition where sudden onset obsessive compulsive symptoms and other neuropsychiatric symptoms all of a sudden manifest in a child who often before was totally okay. And literally sometimes overnight, their personality changes and they develop really terrible OCD and neuropsych and anger and symptoms, making it obviously incredibly difficult for them to manage. And why this happens is still under study. It's considered an autoimmune disease, and there have been certain, again, autoantibodies that certain teams have identified in patients with PANS pandas, including, for example, against dopamine receptors. But at the same time, PANS pandas is known to be associated with a strep infection. That's one of the onsets of these PANS panda symptoms, which is a bacterial infection. And many PANS pandas children also test positive for Bartonella. So there seems to be a combination between strep, Bartonella, and certain autoantibodies. And I will put money on it, and it's one of the paths we're going down, that there are some molecular mimicry relationships between the proteins that Bartonella, even some of the other, independent strains of Bartonella, strep, and some of these other pathogens that are very common in PANS pandas kids, and some of the receptors sort of targeted in the brain of PANS pandas kids that are probably part of the PANS pandas disease process. So yeah, I think you just basically brought up one of the more important areas of research, which is better understanding the capacity for, at the very least, Bartonella to contribute to molecular mimicry-based types of autoimmunity.
I would also just point out very, very quickly that there are many forms of autoimmunity ranging from hemolytic anemia to sarcoidosis that have been associated with Bartonella infection. Because, Bartonella infection, is often sort of precipitated by contact with cats, especially stray cats and very young cats. And so we do know a little bit about it. As Amy mentioned, there's so much more research that needs to be done because these tick and vector-borne illnesses are often very under-researched. But on the books for sure is a link between well-known autoimmune conditions and Bartonella infection chronically in adults. There are many, many case studies and case series of this occurring. So understanding the mechanisms behind why is really important. But the short answer to the question, which is a great question, is yes.
Yeah, it's actually true.
Between Bartonella and autoimmune conditions.
I agree with you. Exactly. Out there are already examples of people who have an autoimmune disease also testing positive for Bartonella, and that's known. Again, though, the why, the exact mechanism of action, which is my favorite part of understanding anything, that still remains to be elucidated more. But yeah, just a very, an exciting area of research to move forward, I think, that can help us probably understand more of what's going on with even especially neuropsych type manifestations.
Yes, I can attest to experiencing neuropsychiatric manifestations during long COVID, and it's it's truly disrupting to everyday life. And really looking forward to getting some answers there, because as I was speaking about my own experiences with these psychiatric manifestations in the public sphere, many people reached out and said, I didn't realize that this could be a part of, um, my, or related to my COVID infection.
Yeah, exactly.
I've even had psychiatrists or therapists reach out and say, um, I'm having so many more patients, like, or clients coming in talking about that they have anxiety, depression, and they were asking me, that is this related to COVID in any way? And I'm like, well, there's definitely a possibility. There are links that have been demonstrated, so
Yeah.
definitely something to keep in mind. So we have some very specific questions here, and I just want to preface by saying we are not offering any medical advice here. This stream is strictly for educational purposes, but we have Christina who asked, they had their strep autoantibodies tested, and they were very high. Did COVID trigger that? And we have no way of knowing for sure, but given what we just talked about, Amy, what would you say?
Yeah, I think it's reasonable, again, to clarify the mechanisms of autoimmunity. There's the bystander activation that both of you mentioned before. It includes the possibility that someone can get an infection, and then what happens is the immune system is targeting the infection, but there's so much inflammation and there's so much cytokine release and other molecules that are part of getting caught up in this, that they might activate T cells or other immune cells that are nearby that weren't actually supposed to be part of this more specific targeting, and then they become problematic as well. I mean, under those conditions, you can definitely have, it's like a bit of instability there as a lot of this fight goes down. You can have instances where, the immune system that was previously maybe not targeting your strep or something like that goes after it. And then again, as long as strep has, either a type of mimicry human target or something like that, you're going to get it. You're going to get it, an antibody generated that's going to, or autoantibody that could, antibody that's,
there was a typo there. Yeah.
If you're, I feel like the way I see it is it's probably a strep antibody. So your antibodies to strep are going up, but that again is a really, it's a liability for those antibodies now to potentially hit human structures or other things with a similar-ish size and shape, and which could lead to more of an autoimmune problem, right? So there can be this, there is this that's obviously a phenomenon that we're looking at in long COVID and related conditions, is like one infection and the immune response to that infection can lead to this immune response situation that could sometimes, become a situation in which pathogens can reactivate or antibodies are produced that then can target others. It's like, so each infection does become an opportunity for other pathogens that you harbored and surrounding immune issues to sort of kick up. So I absolutely think there could be a connection. And it's, again, another one that we have to study more. A lot.
I just want to point out, because Christina added to the question, mentioning that she also had reactivated EBV. Christina, what you're describing here is something that is a common clinical picture in people with long COVID, and often a common clinical picture in people with ME/CFS and chronic Lyme disease and chronic tick and vector-borne illness, where, and we've been talking a lot about autoimmunity, and I also think we also need to sort of address chronic inflammation that occurs as well, which is, often related but different to autoimmunity. And what, again, to sort of cite the work that we've done with Akiko at Yale, we did publish a paper showing that many people with long COVID have reactivated latent viral infections and latent bacterial infections, and we see evidence of these reactivated illnesses. Now, the one thing that we're not 100% sure on, and this goes right back to some of other conversations as well, is that the current state of the testing is such, it is so, just not adequate right now, that when we see antibodies, we can't tell you that you actually have reactivated EBV. Well, in some cases we can, but with the standard first-tier testing, we often can't tell you, do you have reactivated strep? Do you have reactivated EBV? Or does your body just think that that's what's going on and it's producing antibodies for reactivated strep or reactivated Epstein-Barr virus? And that's a very important distinction that we hope that one day testing will occur where we can actually test for elements of the pathogen that make it absolutely beyond any shadow of a doubt, yes, this pathogen has reactivated, versus actually your body just seems to be reacting as though the pathogen is there, but it hasn't actually, reactivated. And I think that this is a really important area of research as well as technology development that has to occur sooner rather than later, because, currently this is something that we're really struggling with, and certainly we see a lot of people with infection-associated chronic conditions who test positive for everything, and they, as well as our clinicians at the center, are sort of scratching their heads, saying, "Well, what does this mean? Are you actually currently reactivated with everything, with all of these viruses, or is there something else going on with your immune system that could be causing problems?"
Ooh, thank you. And we have some more great questions here. Oops. How might microclotting pathology be involved in autoantibodies?
Yeah, there is a lot that we have to learn about the mechanisms between microclotting, but I will hand over to Amy on one of these mechanisms for sure, to do with the actual nature of microclots, as it stands. But there are many ways that microclotting pathology it could be related. I mean, the first and most simplistic way is that a lot of, when we talk about platelet hyperactivation and microclots, a lot of that can sometimes stem from what we call endothelial dysfunction, which is actually pathology of the inside of our blood vessel. And so, probably, one of the simplest ways that autoantibody production and autoimmune behaviors occurring in the body could lead to microclotting pathology would be that these autoantibodies are actually attacking the endothelium. So they're actually attacking cells that are, part of your blood vessels, which then leads to inflammation, which then leads to formation of these small clots, because a inflammatory protein called fibrin is holding together these clots, as well as, the platelets becoming hyperactivated and becoming, highly active as a result of the inflamed endothelium being caused by autoantibodies. That's one very simplistic mechanism, but I also think that functional autoantibodies and the presence of autoantibodies could just in and of themselves, in their, just by the fact that they're present, be responsible for formation of microclots. And I know that Amy, has more to say on that front.
Yeah, the most straightforward way that I see microclots forming is just in response to the spike protein. I think that if you look at the Doug Kell and Mauricio Pretorius team who first characterized microclots in long COVID, they did an experiment where they took the S1 protein created by SARS-CoV-2 and just dropped it into a platelet poor plasma, like a type of blood, and watched a form of fibrin like the microclots form. So in just the most straightforward way, someone getting COVID, spike protein being created by the virus could lead to the formation of a microclot. I actually see it as the most straightforward mechanism. And, in long COVID, we're seeing persistence of SARS-CoV-2, a failure to clear the virus in certain studies. And we see, at least in most teams there's some teams that have sensitive technologies that can measure the spike protein in long COVID blood over long periods of time. In one case, I think one study even 14 months after initial infection, right? And spike protein is still found circulating in the blood of at least a portion of long COVID patients in every single study that's looked at it, right? So if you still have circulating spike and spike can seed a microclot, again, that's kind of the most direct mechanism I see. I think that Riccia Pretorius and her team, though, think that as the fibrin of the microclot forms, maybe there's an opportunity for certain autoantibodies that might, again, have been generated, perhaps even just transiently, though, as part of the inflammatory situation happening with acute COVID, to get trapped inside the microclots. In other words, as part of the fibrin forming, maybe certain autoantibodies that would otherwise have sort of dissipated actually get trapped inside the clots. And there they actually maybe are retained, right? So maybe, the clots themselves could become, I think that's a little bit more of what they've been looking for, is seeing if they can find autoantibodies inside the clots potentially trapped there, which would be, again, like a lot of the way that in human disease, pathogens or autoantibodies created in relation to pathogens persist is by hiding inside things. Like they hide inside immune cells, they hide inside platelets or our white blood cells. Same, like if an autoantibody is created but it's trapped inside a clot or something, again, it's just ways that pathological things can kind of live on, as long as they're not sort of free floating where they're more likely to be cleared. So that can be part of maybe the relationship as well.
Thank you to you both. And on that note, we did cover persistent pathogens in a previous live stream. So please visit our YouTube channel, youtube.com/CoreSinai, CoreSinai, and check out our content there. And someone, to wrap this up, left a really nice note. And I would just like to acknowledge everyone who is doing work, not just people at Mount Sinai, but everyone across the board or around the world, who has focused their attention on solving these problems and getting treatments and solutions to people who are suffering from complex chronic illness. We also thank them, and thank you, Max, for your beautiful comment. It is an honor to do this work and very rewarding to serve. So yes, thank you, David and Amy. We have these live streams monthly on Tuesdays. So which Tuesday kind of depends on our schedules. But right now we are usually doing them on the third Tuesday of every month at 2 p.m. Eastern. But you can follow us on our socials to keep up with us and know when we will be live and answering questions and sharing what we know. Thank you for watching. We've had right now 1,078 people on. So thank you to all of you. And we will see you during the next stream. Bye.
Thank you, everyone.
Bye. Thank you.
Bye.